us in with interesting and learning episode of B.Pharm on the important unit of 3D QSAR first, which are very important units of the section of medicinal chemistry 7th semester, which is prepared by Dr. Vivek Kumar Vyas, Assistant Professor in Department of Pharmaceutical Chemistry, Institute of Pharmacy, Nirma University, Ahmedabad. So let's start our episode while taking a look at what we are going to learn today. Module 1, it is a drawback of 2D QSAR and need of 3D QSAR. Module 2, sources of hits. Module 3, combinatorial libraries. Module 4, selection of lead. Module 5, optimization of lead. And last module is module 6, drug targets. So let's start module 1, drawback of 2D QSAR and need of 3D QSAR. So let's start with the limitation of 2D QSAR analysis. 2D QSAR is a very simpler, faster and more amenable to automation than 3D QSAR method. 2D QSAR include well-defined physiochemical parameters and are best suited for the study of large number of compounds. However, 2D QSAR suffers from some limitations in some cases. In 2D QSAR, two-dimensional structures of molecules are considered. Sometimes in 2D QSAR, compounds are unfit for analysis due to absence of suitable physicochemical descriptors. Only few parameters like steric parameters ES and electronic parameters Hammett constant is used for describing drug receptor interactions. Only those compounds which belongs to congeneric series are considered for 2D QSAR study. Stereochemistry is important for governing the activity of molecules is not considered in 2D QSAR analysis. Sometimes there is a high risk of chance correlations and also too for predictions. No graphical representation of results in 2D QSAR. 2D equation will not rightly suggest new compounds to design. In order to interpret the equation, one should have knowledge of substituent constraint in physical organic chemistry. 2D QSAR methods are based on graph theoretical indices. 3D QSAR analysis is based on the descriptors derived from 3D representation of molecular structures. 3D QSAR become gradually popular with the development of fast and accurate modeling methods for the generation of 3D conformations and alignment of chemical structures. The most popular 3D QSAR methods is comparative molecular field analysis, which was developed by Kramer et al. 3D QSAR is one of the most popular drug design method in the area of ligand-based receptor modeling. 3D QSAR combines classical QSAR analysis and 3D ligand alignment into a powerful 3D QSAR tool. 3D QSAR has been successfully applied to many diverse classes of chemical compounds. Different QSAR techniques have their own strengths and limitations. 3D QSAR methods generally result in the colored maps of steric and electrostatic fields, which can be easily interpreted in terms of specific interactions important for the ligand binding to the receptor. On the other hand, 2D QSAR methods require no conformational search and structural alignment. Thus, in 2D interpretation of the resulting models in familiar chemical terms is frequently difficult, if not impossible. Then come to module 2, sources of hits. Hit is a compound which shows the desired biological activity in the compound screening methods and whose activity assay. Hits identification methods range from traditional screening method to affinity selection of large libraries. Fragment-based techniques and computer-aided de novo design. Heat can be obtained mainly from three sources. The first one is the natural world, which includes plants such as flowers, trees, bushes, roots, branches, etc. Microorganisms such as bacteria and fungi, animals such as frogs, snakes, scorpions, biochemicals such as neurotransmitters, hormones, and marine chemistry such as corals, bacteria, fish, etc. The second rich source of heat is from the synthetic world, which include traditional synthesis of chemical compounds and combinatorial synthesis, and the third one is the virtual world, which include computer-aided drug design. Various screening methods exist to identify heat molecules. High throughput screening method is one of them, which involves screening of compound library directly against the drug target, such as cell-based assay. The activity is dependent upon the drug target but is required secondary assay to confirm the mode and site of action of compounds. Fragment-based lead discovery can also be applied in order to identify heat molecules. Affinity screening techniques are also used, which can accommodate small molecules mixture formats and therefore can increase throughput and sampling or drug or fragment-like molecules. Hits can be obtained from many sources, but eventually a sample of the hit structure must show the activity in a biological assay. And come to module 3rd, combinatorial libraries. Combinatorial libraries are collections of chemical compounds, small molecules or macromolecules such as proteins, which are synthesized by combinatorial chemistry, in which multiple different combinations of related chemicals species are reacted together in similar chemical reactions. Chemical synthesis as a method are used to synthesize large number of compounds. Many advancements in combinatorial library synthesis, purification and analysis techniques are made. This also includes large numbers of compounds which can be tested against a specific biological target and also speeding and improving the overall processes of lead identification, optimization and discovery. There are two main methods of preparation of combinatorial library. First one is parallel chemical synthesis and second is split and mix chemical synthesis. These approaches can apply solid or solution-based synthetic methods alone or in combinations. Majority of combinatorial library synthesis is done on solid support. In a parallel chemical synthesis, all the products are assembled separately in their own reaction vessels. The split and mix approach depends largely on solid-based synthetic methods and produces a mixture of related chemical compounds in the same reaction vessel. A combinatorial library are worthy when they screened against the desired target. The screening of combinatorial library for desired activity depends on the form in which the compounds are present in the library. Pharmaceutical industry uses blind approach for the synthesis of combinatorial libraries on solid phase without either an extensive program to validate the applicable chemistry. The design, synthesis, characterization and screening of combinatorial libraries in biological assays require a huge computational information and management challenge. Potential of combinatorial chemistry and chemical synthesis technology has advanced to the stage where numerically large quality combinatorial libraries at a practical scale of synthesis can be prepared. Dear friends, up till now you have gained a complete idea about drawbacks of 2D QSAR and need of 3D QSAR, sources of hits and combinatorial libraries. If you have missed any of the module or content, you may log on to our website www.cec.nic.in to download videos, frequently asked questions, LOR and other contents so that you can be remain updated with this chapter. Now, let's move ahead to our next module, that is about selection of lead. Module 4, Selection of Lead. Selection of the right lead compounds for drug development is a very important step in the drug discovery process. Many potential drug candidates fail at late stage of drug discovery due to lack of efficacy or unexpected adverse events. Lead selection is a very important and crucial step in drug discovery and development. A lead compound or substance is one that is believed to have potential to treat disease. Laboratory scientists can compare known substances with new compounds to determine their likelihood of success. Leads are sometimes developed as collections or libraries of individual molecules that possesses properties needed in a new drug. Lead identification usually involves high throughput screening in which large libraries of chemicals are tested for their ability to modify the target. This process enables researchers to conduct millions of biochemical, genetic or pharmacological tests and rapidly identify those that modulate a particular biomolecular pathway. These screening tests are usually simple in vitro binding assays. Once all the hits are identified, then the selection process starts for ranking chemical series for the selection of lead compounds. The key considerations might be radically different for hits derived from the different hit discovery techniques. For hits from high throughput screening, selectivity, chemical tractability, binding mechanism, pharmacokinetic properties and freedom of operation are usually observed as more significant than the actual potential of the identified hits. In case of fragment-based hits, there is a generally little value in judging selectivity for weak hits originating from small and diverse fragment libraries. The availability of structural information on binding mode and opportunities to rapidly and easily produce structural analogues might be highly valued, whereas pharmacokinetic or intellectual property evaluation often makes little sense at such an early stage. Lead selection methods are also dependent on the screening technology used for hit discovery. Lead selection process for drug-like hits involve techniques such as hit evolution, bioisosteric replacements and hit fragmentation or any combination of these. Hit evolution is one of the most frequently used techniques for selection of lead when analogues of the original hits are synthesized with different substitution patterns and screened for desired activity. Combinatorial libraries created using solid or solution phase parallel synthesis coupled with high throughput screening to facilitate compound output. Crude or purified compounds synthesized in mixtures can be screened for desired activity under with affinity screening. Structure-activity relationship data obtained after screening of compounds libraries can then be used to drive experimental medicinal chemistry efforts, producing compounds with an improved profile, and it is called lead-like molecule. Bioisosteric replacements represents a case of hit evolution when hits are transferred using bioisosteric rules. Successful lead selection from high throughput screening hits resulted in drug-like leads regardless of the lead likeness of the original hit. Problems in the lead likeness of the hits are basically attributed to molecular weight and log P, reflecting the fact that hits are often large lipophilic molecules. Come to Module 5, Optimization of Lead. Optimization of lead is phase to maintain desirable properties in lead compounds while improving on deficiencies in the lead structure. Lead optimization is the complex, non-linear process of refining the chemical structure of a confirmed lead to improve its drug characteristics with the goal of producing a preclinical drug candidate. This stage frequently represents the bottleneck of a drug discovery program. Lead optimization compares the properties of various lead compounds and provides information to select the compound or compounds with the greatest potential to be developed into safe and effective medicines. This can involve similarity searching to find compounds previously made or available commercially for purchase. To assess similarities, the following properties are analyzed: absorption, distribution, metabolism, excretion, toxicity. The testing of analog series results in quantitative information that correlates changes in chemical structure to biological and pharmacological data generated in later stages. Based on these results, medicinal chemists make specific changes to the lead compound, which they hope will improve its binding affinity in order to optimize pharmacological properties such as bioavailability or stability. At that point, the new analog feed back into the screening hierarchy for the determination of potency, selectivity, and mechanism of action. These data then feed into the next optimization cycle. The lead optimization process continues for as long as it takes to achieve a defined drug profile that warrants testing of the new drug in humans. When testing a large number of compounds in order to identify a new lead, it is best to have compounds that are as different from each other as possible. Pharmaceutical companies purchase large number of compounds from third-party suppliers. They also synthesize combinatorial libraries of compounds. Chemical diversity is an important feature of such compound collections and libraries, hoping to cover as much chemical space as possible. Often during the same stage of development, lead prioritization studies are conducted in living organisms and in cells. The test tube to compare various lead compounds and how they are metabolized and affect the body. Lead optimization process concerns many aspects such as the optimization of the affinity for the biological target, the toxicity, the oral bioavailability, the cell permeability, the plasma binding, and the ease of metabolism. The principle employed is that any incremental change in the chemical structure produces incremental changes in bioactivity, and a systematic study of such cause and effect relationship is called structure-activity relationship study. The process is highly iterative and traditionally based on trial and error. This is a stage in which the various elements that constitute further characterization are carried out will vary from company to company, and parts of this process may be incorporated into the lead optimization phase. All the information gathered about the molecule at this stage will allow for the preparation of a suitable candidate, which will, together with toxicological and chemical manufacture and control considerations, will form the basis of a regulatory submission to allow human administration to begin. Once the lead is optimized, the attrition rate of compounds entering the clinical phase is also high. Again, only 1 in 10 compounds reaching the market. Then come to Module 6, Drug Targets. Drugs usually act on either cellular or genetic chemicals in the body known as targets, which are believed to be associated with disease. Scientists use a variety of techniques to identify and isolate individual targets to learn more about their functions and how they influence disease. Compounds are then identified that have various interactions with the drug targets that might be helpful in treatment of a specific disease. Drug target is a very broad term which can be applied to a range of biological entities. Drug targets can be proteins which include receptors, enzymes, carrier proteins, structural proteins like tubulin. It can be cell membrane, lipids. The drug target can be nucleic acid which include RNA and DNA, and it can be carbohydrates which includes cell surface carbohydrates, antigens and recognition molecules. Selection of drug targets is very useful in the development of new drug candidate for the treatments for disease. Researchers analyze and compare each drug target to others based on their association with a specific disease and their ability to regulate biological and chemical compounds in the body. A druggable target is available to drug molecule which can be a small molecule or larger biologicals, and upon binding with the target produce a biological response which can be measured both in vitro and in vivo. Certain targets classes are more open to small molecule drug discovery. For example, G protein coupled receptors, whereas antibodies are good at blocking protein interactions. Tests are conducted to confirm that interaction with the drug target are associated with a desired change in the behavior of diseased cells. Research scientists can then identify compounds that have an effect on the target selected. Drug target identification and validation allows increased confidence in the relationship between target and disease and permits us to explore whether target modulation will lead to mechanism-based side effects. More recently, chemical genomics, a systemic application of tool molecules, has emerged as a good technique to drug target identification and its validation. Now come to conclusion. 2D QSAR suffers from some limitations like two-dimensional structures of molecules are considered. Compounds are unfit for analysis due to absence of suitable physicochemical descriptors, and congeneric series are considered for the study. Stereochemistry of the molecules is not considered and no graphical representation of results in 2D QSAR. 3D QSAR analysis is more advanced method and now become gradually popular with the development of fast and accurate modeling methods for the generation of 3D conformation of the molecules. Comparative molecular field analysis is the most popular 3D QSAR methods. HIT is a compound which shows the desired biological activity in the compound, screening methods and whose activity assays. Three main sources of hits are the natural worlds, synthetic world, and third one is the virtual world, which include computer-aided drug design. Combinatorial libraries are collections of chemical compounds, small molecules and macromolecules synthesized by combinatorial chemistry. Chemical synthesis methods are used to synthesize combinatorial libraries. Many advancements in combinatorial library synthesis, purification, and analysis techniques are made. A lead compound or substance is one that is believed to have potential to treat disease. Lead optimization is the complex, non-linear process of refining the chemical structure of a conformed lead to improve its drug characteristics with the goal of producing a preclinical drug candidate. Drugs usually act on either cellular or genetic chemicals in the body, known as targets, which are believed to be associated with disease. If you have missed any of the module or content, you may log on to our website www.cec.nic.in to download videos, frequently asked questions, LOR and other contents so that you can remain updated with this chapter. Now, let's move ahead to our next module, that is about selection of lead.